So transcription and translation, remember the two processes that link gene to a protein. The process of rewriting or copying the gene, and then reading that gene and turning it into a protein. So we're going to go through the process of transcription. We're going to take a pre-mRNA transcript, which is a result of that. We're going to process it and do some things to it, and then we're going to translate that into a protein inevitably. So each of those steps we're going to go over in detail. So in a eukaryotic cell, we go from DNA. We then make a copy of that DNA through the process of transcription, right? We're rewriting. Transcription means to rewrite. We then actually, in a eukaryote, because we're more complicated, right, than a prokaryote, we have to do some RNA processing. We process the RNA before we actually, before it's actually read at a ribosome. We're going to talk about how we do that and the purpose of the things that we do in order to modify that RNA before it can be read. And then we translate it. Notice where transcription and RNA processing happen. They happen in the nucleus. Translation happens in the cytoplasm. Okay, you probably have no idea who that is, but it was funny, I promise, back in the day. But look it up. It's an SNL skit with, what's his name again? I can't think of it. Rob Schneider. Yeah, right. So the three stages of DNA synthesis or of RNA synthesis of transcription, which is RNA synthesis. Remember, this is different than DNA synthesis. It is initiation, elongation, and termination. Initiation is the start of the process, elongation is the actual copying, and the termination is the stop of the copy. So the process of initiation, there's actually a promoter site in a gene. Okay, so a sequence of DNA we want to copy. Upstream of the actual message itself is something called a promoter. And the way we can tell a promoter is it has this sequence of TA, TA, repeating, and we call it the TATA box. The TATA box is just part of the promoter that can be recognized that this is the beginning of a gene. So you look for a TATA box, and that's where the promoter is. Then an enzyme called RNA polymerase. Remember, DNA polymerase copied DNA. RNA polymerase copies RNA. Then what happens is there's these special proteins that will attach to the TATA box. Think of them as gripping points for RNA polymerase. If they're not there, RNA polymerase can't bind. Once the transcription factors bind to the TATA box, and these transcription factors are things that can be like turned on during signal transduction or something, so then they'll go to the TATA box, they'll bind after a ligand binds to a receptor, you have your phosphorylation cascade, inevitably turning a gene on. They become transcription factors. Same thing with steroid signaling, right? A steroid binds to a receptor on the inside because it can cross the membrane easily because it's nonpolar. And then that becomes a transcription factor, and that will go to a TATA box, or the promoter of a gene, and turn that gene on. Sometimes you have transcription factors that'll turn the gene off or block it from binding. So RNA polymerase and transcription factor together become the transcription initiation complex, and that's going to cause the DNA to unwind. Yes, bud, what's up?
Can you make some hot chocolate for me?
You want some hot chocolate?
Yeah, I'm gonna watch.
What?
Well, who are you talking to?
I'm recording. I'm actually recording this for my class.
Oh.
Do you want me to make you hot chocolate?
Mhm.
All right, time out. Hot chocolate time. All right, I'm back. So, the enzyme that does this is called RNA polymerase. It's the main enzyme that drives transcription. It makes a polymer of RNA, right, a long chain of RNA. Remember, just as a review, anytime you see the suffix ASC, that means enzyme. Not all enzymes end in ASC, but if you do see that, that means it's an enzyme that drives some sort of chemical reaction. In this case, right, it's a long chain of polymers. We put them together. So this is an anabolic reaction. It is endergonic, and it is non-spontaneous. So this is showing you a sequence of DNA, a gene, right? So this is not RNA yet. And here you can see the TATA box up top and the promoter of the gene. The DNA template, this starting point, this is the actual recipe that we're trying to copy. So here's our promoter ahead of time and what we actually want to copy. Then you see a bunch of transcription factors, which are these special proteins that bind to the TATA box. Once they're all bound, finally RNA polymerase, which is this teardrop-shaped protein or enzyme, can bind. And once that happens, it will now slide along the sequence of DNA, making a copy off of the template strand. Notice the template strand is on the bottom. And the template strand tends to run 3' to 5' because we are again going to synthesize our new RNA strand just like we did with DNA polymerase. Our RNA polymerase will synthesize in the 5' to 3' direction. Then elongation is essentially the RNA polymerase sliding along the DNA, untwisting it, exposing about 10 to 20 base pairs of RNA at a time, or of DNA at a time for RNA nucleotides or ribonucleotides to bind to it, right? So it adds to the 3' end, or it grows 5' to 3', just like a DNA polymerase did. And as it slides through, the DNA helix will now just reform itself on the other side of it. I'm going to show you guys a video of what that looks like in a second. And it does this about 60 nucleotides per second in eukaryotes. That's the rate. And so here's showing you how see how it peels apart the DNA, and then it synthesizes, which the DNA is in the blue, it synthesizes new RNA. Notice the U's, and that's that pink strand right there. And it's doing it in the 5' to 3' direction. See, 5' synthesizing on the 3' end. Then once it gets to the end, we have to terminate it. Okay, so it hits something called the terminator sequence in the DNA. In prokaryotes and eukaryotes, it's different. Prokaryotes stop the termination. It's a specialized sequence, but the eukaryote will hit a signal of poly A, like long chains of A's, A, and it keeps going, and then we'll cut free of the DNA. Okay, you don't have to really know that. They're not going to ask that on the AP Bio exam, but that's how it stops the process. It gets terminated. So here's showing you the whole thing. You can see the RNA polymerase. Now notice they're not showing the transcription factors binding just for simplicity. That does have to happen first. It's kind of like, remember how we had a primer for DNA replication? And this, it's kind of like the primer, but instead the transcription factors, the proteins have to bind the promoter. The RNA polymerase will then slide along until it hits a terminator sequence. Once it hits the terminator sequence, right, here's it sliding along in elongation, it hits the terminator sequence and it falls off. And now we have our copy of our RNA. This is, remember, that disposable copy of the gene that we want to read and turn into a protein eventually. Okay, so you can kind of summarize transcription with a drawing in your notes if you'd like. If you get it, you don't have to, but I'd recommend at least trying to think about it to make sure that you understand all the different parts, both initiation, elongation, and termination. Now, the next step to this is eukaryotic cells actually modify their RNA at transcription. After transcription is over. Prokaryotes won't do that. So that initial transcript that is produced is called pre-mRNA. It's not actually finished yet. So we have to modify it. And this process happens in the nucleus, and there are 3 different things that have to happen. We have to add a 5' cap and a poly A tail. Both of those slow down degradation, and they actually, so because RNA actually gets broken down eventually. It's kind of a way that we control and regulate this process. So the 5' cap helps to stop it from being degraded, helps to stabilize it. And the poly A tail almost acts like a long tail. The longer it is, the longer that the RNA can remain before it gets degraded. So it kind of acts like almost like a timer on it for how long it can stay in the cell before we break it down. And then the last, and I shouldn't say the most important, but a very important step that we have to do is we do something called RNA splicing, which is where we cut out sequences of the DNA that actually don't code for anything, or excuse me, sequences of the RNA that don't code for anything in the pre-mRNA transcript. We'll get to that in a second. So the 5' cap and poly A tail are added to different ends because they'll add stability and they'll lower the chance of degradation of the message. Okay, you want to know that. It's something that could assess in the AP Bio exam. Here's showing you the 5' cap, the poly A tail, and then the coding segment of DNA. This is going to be what actually is read to make a protein out of. That is like the recipe. The third thing that happens is RNA splicing that I mentioned. All right. There are actually lots of nucleotides that are not needed in the eukaryotic genome, and we don't know exactly why this evolved entirely. It could help to buffer against mutations, because if we have non-coding sequences, that sequences is not spelled correctly. But those sequences would be able to gain mutations and be mutated, where we kind of buffer that against the other ones. That they're actually coding. But when we actually want to code for the protein, we have to cut them out. The non-coded segments, and you have to know this, are called introns. So we want to cut out the introns. They're gibberish DNA. Again, we don't really know why we have them. There's some hypotheses, as I said, but they don't know. Um, the ones that are expressed are the exons. Do you want to remember that? Sometimes people get this confused and they think the exons are cut out because it means like, like XIs or, but that's not what exon means. It means expressed. Okay, where an intron is not expressed. We want to cut those things out. And we do this through these special types of structures called spliceosomes and small ribonucleotide proteins and RNA, special RNA molecules that will cut these introns out. So you can see here are the exons separated by introns or non-coding gibberish DNA. We cut those introns out, we put the exons together, we glue them back together, and now we have our final mRNA that can be read. We have our 5' cap, our poly A tail, and the introns have been removed through splicing. This shows you the spliceosome, which has the small nuclear RNAs here. Okay, it's part of in the protein that helps to attach to the introns, and then it basically cuts out the excised part, or it excises this part out, and then sticks our exons back together for our coding protein. Now, a very important thing that you want to understand is that so protein has multiple exons, and we can do a process called alternative splicing, which can actually create different proteins from a single gene. Notice this gene has 3 exons in it. Well, we don't have to put every single domain, as you can see below. See the purple, the yellow, and the green domain. We don't have to put them all together to make a protein that's functional. We could also maybe just put domain 3 and domain 2 together, or domain 1 and domain 3, or 1 and 2. You can do all these different things. So that's another reason they think introns could exist, is it allows us to create more complex proteins from just in one gene. Okay. So we have transcription, RNA processing, translation, and usually, as I said, each of these exons codes for some important component of the protein itself. So like maybe the first part, like let's say this is an enzyme, maybe domain 3 is a regulatory site where an allosteric inhibitor or activator can bind. And maybe domain 1 is the active site where the substrates will actually bind. And maybe domain 2 is more of a structural unit to just give it support and structure. So it just depends. But usually each exon kind of has its own little role. And this is what I mentioned before, is that we can actually do this alternative splicing, and this is kind of showing a simplified version of that, where we can actually reorganize different exons together, splice them together in different ways to create different proteins from one gene. And that's again why I said one gene, one protein, or one gene, one polypeptide is not technically correct. What's really nice is prokaryotes, though, are much simpler. They do not have to modify their RNA at all. Their transcripts are directly turned into proteins directly. Directly turned into proteins directly. They are turned into proteins directly. They don't have to modify them. Okay. Okay, so now we've transcribed a gene and modified it. What do we now have to do? We now have to turn that RNA into protein. Okay, and that's called translation. The process of taking a nucleic acid, RNA, and turning it into a protein, amino acids, right, is changing languages. But we have to know some information about the message first before we can actually talk about the process of turning it into proteins. All right, so the RNA itself, the message, is actually organized into these little readable units called codons. They're these like 3-letter words basically, right? So there's, remember, 4 different nucleotides you can have, A, U, G, and C, because it's RNA. There's no T's, remember. And they're like the words of an essay. The ribosome has the ability to actually read those words, those 3-letter words, and each word codes for different amino acids. Just as words code for ideas in our mind, each of those codons codes for an amino acid specifically. Sometimes there's more than one word for each individual amino acid too, which we'll get to. But that's essentially the way this works. So a codon, as you can see here, is a RNA nucleotide that's 3 bases long, like GCU, ACG, GAG, and they're like words. The first codons were deciphered in the 1960s, so not long after the DNA molecule structure was figured out, which is really cool. And the genetic code, which is the language, people confuse this. they sometimes think, oh, your genetic code is your DNA. No, the genetic code is the language that every single organism uses to read these words. It's kind of like a Rosetta Stone or something. It's how every organism on this planet reads the genetic code, or reads the DNA or the RNA in the exact same way. And these nucleotide triplets will specify individual amino acids. Now there's 4 nucleotides, as I said, A, U, G, and C. There's how many different possible combinations? Well, if there's 4 nucleotides and there's 3 positions, if you do, your probability math there, you get 64 possible combinations of 3 with 4 nucleotides. And we actually synthesize 20 different amino acids. So that means each amino acid must have more than one word associated with it. And that's called being degenerate, or redundant, because we don't have 64 different, we have 64 combinations, but when they have 20 amino acids, so we have multiple words that code actually for the same amino acid. So how many nucleotides would be needed to code for a polypeptide with 100 amino acids? Well, at minimum, we need 300. Right? We need 300 RNA nucleotides, because for every 3 nucleotides, you get 1 amino acid. And they like to ask that question on the AP Bio exam too. I've seen that. Okay. All right. So the way they figured this whole thing out, I'm just going to breeze through this real quickly, is they actually created long chains of like the same letter of poly U over and over and over again. They figured out all 64 by the mid 60s. So it only took less than 5 years in order to figure out the entire genetic code, which is pretty amazing. This was the first one, was phenylalanine that they discovered. Phenylalanine is an amino acid. And then the polyUs, right? That's it. So UUU is the word that codes for phenylalanine, the amino acid. Remember, amino acids have that amino group, the R group, and the carboxylic acid group next to it. 61 code for amino acids, and 3 are called stop codons. That's how the ribosome knows to stop assembling the protein at that point. There's also a nucleotide, or a codon called AUG. This is one you have to remember. AUG has 2 functions. It actually codes for methionine, which is an amino acid, but it also is a start codon. It signals the beginning of a gene. So the first AUG you find on an mRNA is going to be the start codon, and that's where the ribosome knows to start reading and go every 3 after that point. So this is, again, they're not going to ask about this, but this is how they discovered at least the first codon. They took poly-U's, long chains of U, ribonucleotides, and they ended up getting just phenylalanines in a long chain from poly-U. So they know UUU coded for phenylalanine. Then they created all these different ones, all these different combinations of different ribonucleotides, like UUG, 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 and they started coding for different things, and that's how they figured this whole thing out. So they won a Nobel Prize for figuring this whole thing out because of how important it was. So here's showing you kind of the idea. We go from DNA, here's our transcriptional unit, our mRNA, right? They don't show it being modified, right? But it was modified beforehand. The introns were removed, and the 5' cap and the poly A tail was put on. And then the codons, the 3-letter words. So this, for UGG, for example, codes for tryptophan. UUU codes for phenylalanine. GGC codes for glycine. UCA codes for serine. Now, you're not going to have to know the language, but you do have to know how to use a chart that teaches you how to read that language. This is that chart. So this would be given to you. So what you do is you look for on your mRNA, you find the first AUG, right? You find your first start. And then you read 3 letters after that. So let's say, for example, it's CCU. Well, what's the next amino acid? CCU is right here. Proline was the next one. Let's say the next one is GAU. You find your first base G, A, here it goes, GAU, asparagine. And you just go in order every 3 until it tells you, until you hit a stop codon, UAA, UAG, or UGA. Once you hit that, then you stop reading it, and that's the end of your message. That's the end of the protein. Remember the genetic code. Remember, the code is not your genes. The code is how do we read the message. This evolved very early on because all life uses the exact same code. And that's super important for things like genetic engineering, because we can take our own genes and put them into other things, or we can put other things into our genes, or we can put genes of um, plants into other plants to help make them more hardy or whatever. That's all part of genetic engineering, as we'll talk about. And there's obviously moral implications of that too. But it's because everyone has the same, or everything on this planet has the same genetic code, that we can do that process. And that's why it's called being universal. So it must have evolved very early on, as all life forms have that same code. So now we know. We have our DNA replication, right? That's that replication fork. We're going to copy the DNA. We have the process of transcription going to RNA, then translation going to protein, both of which processes we've kind of talked about so far. We have not talked about translation in depth yet, but we're going to, how does the ribosome do the process of translation?